In this video, we are going to talk about enzyme inhibition. To better understand how inhibition works, we are first going to discuss how enzymes function in normal condition. The substrate is going to bind to the active site of the enzyme, forming the enzyme-substrate complex, which later leads to product formation. Note that the shape of the substrate is complementary to the active site of the enzyme. Inhibitors are chemical substances that can bind to an enzyme and reduce its activity. Inhibitors can be formed from within the cell or can be introduced from the external environment. In this video, we are going to talk about two types of inhibitors, namely competitive and non-competitive inhibitors. Competitive inhibitors have a similar shape to the substrate. They are going to compete with the substrate to bind to the active site of the enzyme. Imagine the final round of musical chair. There's only one chair and two players racing for it. The chair represents the enzyme active site, the players represent the substrate and the competitive inhibitor. Only one can sit. Whoever gets there first occupies the spot. In the same way, during competitive inhibition, both the substrate and inhibitor competes for the active site of the enzyme. The faster or higher affinity molecules win the spot. If the inhibitor binds first, the substrate is blocked, the enzyme cannot carry out its reaction. But if more substrate is added, it can outcompete the competitive inhibitor. Non-competitive inhibitors, on the other hand, bind to an alternative site on the enzyme which is not the active site. These sites are called allosteric sites. Binding to the allosteric site will lead to conformational changes in the active site of the enzyme, such that the substrate can no longer bind to the active site. Imagine someone is about to play the musical chair game, but before the round even starts, someone comes and cuts one of the chair's leg. The chair is the enzyme, the person sitting is the substrate, the person cutting the chair's leg is a non-competitive inhibitor. In other words, non-competitive inhibitors do not compete for the active site of the enzyme. They're going to bind elsewhere called the allosteric site. This changes the enzyme shape so that the substrate can no longer bind to the enzyme. No matter how much substrate you add, the enzyme won't return to its full function. Manipulation of enzyme function with inhibitors is really important to maintain metabolism and regulate normal cellular function. In this way, inhibitors can act as therapeutic drug to prevent diseases and maintain normal cell cycle and cell growth. However, inhibitors can also have some harmful effect where they can act as poison, herbicide, and pesticide. Cyanide is an example of a competitive inhibitor. Normally, oxygen binds to the active site of cytochrome C oxidase, which is a key enzyme in the electron transport chain responsible for producing ATP. Cyanide competes directly with oxygen for the same active site. When cyanide binds, it blocks oxygen from attaching, effectively stopping the enzyme function. This causes a halt in cellular respiration, leading to a lack of ATP. As a result, a cell cannot use oxygen even though it's present. This causes chemical suffocation. The brain and the heart are most affected because they need constant ATP. Now let's see the example of memantine, which is a non-competitive inhibitor of the NMDA receptor. Normally, glutamate binds to the NMDA receptors to allow calcium ion to enter neurons, which is important for learning and memory. In Alzheimer's diseases, there is excess glutamate activity leading to overstimulation of NMDA receptors. This causes too much calcium influx, damaging neurons. Memantine binds to a different site, the allosteric site inside the NMDA receptor ion channel. This blocks excessive calcium entry without completely shutting down normal glutamate signaling.